In previous class, we already discussed about biotechnology applications. Okay. Briefly, you already know about applications of biotechnology, transgenic plant, pest resistance plants formation, and also other applications of biotechnology. Okay. Now in this class, I want to go to discuss about transgenic plants. Okay. Transgenic plants. What are transgenic plants? It's plants with desirable characters created through gene transfer method. Gene transfer method is known as transgenic plants. In this, different types of methods are involved in this transgenic plant formation. But in this transgenic plant formation, desirable characters are created. Desirable character, wanted characters are created through gene transfer method. This is known as transgenic plants. Okay. In this transgenic plants, different types of methods are involved to introduce the clone genes. Introduced clone genes into the plant cell. The TI plasmid is extensively used in the genetical modifying crop methods. Okay. TI plasmid, it is present in Agrobacterium tumefaciensis. Agrobacterium tumefaciensis. It is widely used in the transgenic plant formation. Okay. It is widely used and also successfully obtained transgenic plants. Okay. The next one, transgenic plants are need of agriculture and human welfare. It is very, very need to agriculture. This means a crop improvement program, disease resistance program. Okay. Next one, high nutrient utilization, high yielding formation. These are different types of properties are obtained from these transgenic plants. Hence, transgenic plants are very, very necessary of agriculture and human welfare. Okay. Several types of transgenic plants have to produce to meet a specific needs of agriculture and human welfare. There are some agriculture and human welfare needs are given this. The first one is the transgenic plants are resistant to pathogens and pests. Pathogens and pests. Some like papaya, carica papaya, bobai. Okay. Some carica papaya plants are transgenic plants are there are resistance to ring spot virus. Ring spot virus. Okay. Some transgenic plants like BT cotton plants are resistance to insects. Insects, caterpillar like insects are resistance these BT cotton like transgenic plants. The next one, transgenic tomatoes. These are also resistant to some bacterial diseases, especially the Pseudomonas bacterial diseases. Pseudomonas bacterial diseases. Okay. Next one, some potato plants are also resistant to some fungal organisms, fungal pathogens like Phytophthora like fungal organisms cause some diseases in potato, but transgenic potato plants are resistant to that type of diseases. Okay. In this way, the transgenic plants are resistant to pathogens and pests. Okay. Transgenic plants are suitable for food processing technology. Food processing technology. This means a transgenic tomato, like a flower saver. Transgenic tomato, this one, flower saver. Okay, this flower saver plant is bruishes resistance. That means it is suitable for storage, transport due to delaying ripening and also long shelf life. Okay, స్టోరేజ్ వాటి స్టోరేజ్ చేయటం నిల్వ చేయటం అనేది చాలా ఈజీ అండ్ ఆల్సో ట్రాన్స్పోర్ట్ ఎక్కువ దూరం ట్రాన్స్పోర్ట్ చేయటం అనేది ఈజీ ఎందుకంటే డ్యూ టు డిలైన్ రైపనింగ్కి త్వరగా రావాలి. అంతేకాకుండా దేర్ ఆర్ ఆల్సో కంటే లాంగ్ సెల్ఫ్ లైఫ్, ఎక్కువ సెల్ఫ్ లైఫ్ ఉంటుంది. హన్స్ బికాస్ ఆఫ్ దిస్ రీజన్ దిస్ ట్రాన్జెనిక్ టొమాటో ఫ్లవర్ షవర్ is suitable for storage and also suitable for transport in long distance. Okay, this one is a very important tomato transgenic plant.
Both of these labs involve the insertion of a new gene into the DNA instead of the deletion or substitution of the gene. The gene of the tomato that is responsible for controlling the length of the life is turned off, and instead a new gene is inserted. This new gene is responsible for keeping the tomato alive for a longer period of time and keeping it firm and fresh for over 2 weeks longer than the original gene would have. The amino acid sequence for the first gene was 210 amino acids long, but the amino acid sequence of the second gene was approximately 270 amino acids long, which clearly shows that additional amino acids were added to help with the tremendous improvement in the life of the tomato. In this image, you can see the effects of the ordinary tomato versus the gene-silenced tomato over a period of 45 days. As you can see, the ordinary tomato rots after 45 days, but the gene-silenced tomato stays fresh. This diagram shows the process to produce tomatoes with double their present shelf life. The scientists isolate the tomatoes with characteristics such as taste, color, and yield. They grow more of the tomatoes to look for the genes. The seeds are then planted and grown for several generations.
Transgenic plants improve nutritional value. Because the transgenic plants are modified to main purpose disease resistance and also tolerant to stresses and also increase food value. Quality food is obtained from these transgenic plants. That's why the transgenic plants are improved nutritional value. You know already golden rice from Taipei. It is obtained from Taipei. Okay, this golden rice is contain rich vitamin A in that food. Rich vitamin A. Okay, this vitamin A is utilized for curing anti-giraffophthalmia. Vitamin A is helpful for anti-giraffophthalmia. This means blindness. Okay, so the golden rice Taipei contains rich vitamin A and also prevents blindness in poor people, because poor people does not consist have no valuable food because economically background, so the valuable food is not borrowed. Okay, hence the golden rice, it is a low cost rice and also provide every poor people to the government. Because of this reason, the Taipei modified golden rice plant is consisted vitamin A. This means vitamin A produced gene is inserted into this Taipei plant. Okay, so if you take this golden rice, you may not suffer with anti-giraffophthalmia. Okay, so the blindness is cured, or blindness is prevented by this golden rice food material. In this way, different types of quality and quantity food material is improved by the transgenic plants. transgenic plants are useful to hybrid seed production. Hybrid seed production, like Brassica nepis. Brassica nepis. It produced the male sterile plants are produced from this Brassica nepis, from especially genetically modified transgenic plants. It is eliminate is emasculation. Emasculation. This means emasculation means removal of anther from bisexual flower. Removal of anthers from bisexual flower is known as emasculation. This type of transgenic plants are produced in male sterile plants. Hence, eliminate emasculation and also reduce the cost of hybridization seed production. So, it is very suitable for hybrid seed formation. Hybrid seed formation, hybrid seed production. Understand? One, the transgenic plants are tolerant to abiotic stresses. Tolerant to abiotic stresses. This means, suppose abiotic stresses, biotic stresses, and also abiotic stresses. Two types of stresses are present. Abiotic stresses means nothing but they are tolerant to cold, drought, salt, heat, some other chemicals, etc., etc. This type of stresses are known as abiotic stresses. Abiotic stresses. Especially transgenic plants are tolerant to abiotic stresses. Okay? This means suppose basmati rice. It is a resistance to biotic and also abiotic stresses. Abiotic stresses. That means drought condition, heat condition, cold condition, chemical condition. Okay. In this way, this type of abiotic and biotic stresses are also tolerant by this basmati genetically modified transgenic plant rice crop. Okay. Next one, soya bean. It is also resistance to herbicidal tolerant. Herbicidal tolerant. Okay. Herbicides ఎక్కువగా పెరిగినా కానీ these plants are tolerant to biotic stresses. Biotic stresses. Herbicidal tolerant. Okay. The type of plants are known as herbicidal tolerant. మన herbicides మనం spray చేసినా కానీ there are also survive like soybean plants. Okay. In this way, transgenic plants are tolerant or resistant to pathogens and pests. Tolerant plants are suitable for food processing technology. Tolerant plants are improved in nutritional value, and tolerant plants are useful for hybrid seed production. Tolerant plants are useful to abiotic stresses. They are tolerant to abiotic stresses. These are the main beneficial aspects of transgenic plants. Okay. So, these transgenic plants are especially alternative resources to industry, such as like starch, fuel, and pharmaceutical industry. This transgenic plants have to shows some expresses gene like insulin, interferon, human growth hormones, antibiotics, antibodies, etc., etc. This type of expressed genes productions are mainly obtained from the transgenic plants. Next one, these transgenic plants are biochemical products are also obtained these plants, and also sometimes better than these produced in bacteria. Bacteria నుంచి ప్రొడ్యూస్ చేసుకునేటువంటి ప్రొడక్ట్స్ కంటే ఈ ప్లాంట్స్ నుంచి ప్రొడ్యూస్ చేసుకునేటువంటి ప్రొడక్ట్ అనేది చాలా బెటర్గా ఉంటుంది. నెక్స్ట్ వన్, utilization of plants are biofactors or bioreactors for obtaining commercial useful products. This means specialized in medicines, chemicals, and antibiotics. These are observed in a large scale from the industrial level. Okay, industrial level. So in this these transgenic plants are very useful to human welfare and also agriculture field. Okay, in this way transgenic plants are very essential to human welfare, very essential to agriculture for green revolution. Understand?
Agrobacterium tumefaciens is a natural genetic engineer of a wide variety of plants. The bacterium has a loop of DNA called a tumor-inducing or TI plasmid and can transfer part of this plasmid into a plant cell. The DNA transfer induces plant cells to make food molecules for the bacterium. Agrobacterium, which is ubiquitous in the soil, begins to communicate with plant cells at a wound on the plant body. The wounded plant releases phenolic compounds that, depending on the species involved, Agrobacterium recognizes. In the cell envelope, a protein called Vir-A detects the chemical signal from the wounded plant. The binding activates Vir-A, which can then activate another protein called Vir-G through phosphorylation. A number of genes called Vir genes are located on the TI plasmid. While the genes encoding Vir-A and Vir-G are turned on all the time, the expression of the other Vir genes requires activated Vir-G as a transcriptional activator. Two of the gene products are Vir-D1 and Vir-D2. The Vir-D proteins are endonucleases that cleave at the 25 base pair sequence repeats at the ends of the tDNA. As a result, the cell produces a single-stranded tDNA molecule attached to Vir-D2 at one end. Vir-D2 directs the transfer of the tDNA into a plant cell in a process similar to bacterial conjugation. Within the plant cell nucleus, the tDNA becomes integrated into the plant genome. A number of genes are encoded in the tDNA and become expressed in the plant cell. Some of the gene products are responsible for the synthesis of the plant hormones cytokinin and auxin, which trigger the plant cells to divide rapidly and form an undifferentiated mass known as a crown gall tumor. The tDNA also encodes genes that control the production of unusual amino acid derivatives called openes. In return for delivering the tDNA to the plant, the bacterium gets the openes as a source of carbon and nitrogen. Researchers employ Agrobacterium to genetically engineer a wide variety of plants. In this procedure, the tDNA is placed on another plasmid that is more easily manipulated, and the DNA between the flanking sequence repeat regions is replaced with a gene of interest. Upon infection, the virulent system induces transfer of the tDNA to the plant cell without any tumor-inducing genes, allowing genomic integration of the recombinant gene without tumor induction or opene production.
Transgenic plants. Introduction of foreign gene. In genetically engineered plant cells, a bacterium, Agrobacterium, is mainly involved in transfer of foreign gene. However, Agrobacterium cannot infect all plants since it has a narrow range of host specificity. Therefore, other techniques have been developed to introduce foreign DNA into plant cells. Novel methods of ensuring DNA uptake into cells include electroporation and mechanical delivery or biolistics. Electroporation is the process of creating temporary pores in the cell membrane by application of electric field. Creation of such pores in a membrane allows introduction of foreign molecules such as DNA, RNA, antibodies, drugs, etc. into the cytoplasm. The development of this technique is due to the contribution of biophysics, bioengineering, cell and molecular biology. While the technique is now widely used to create transgenic microorganisms, plants, and animals, it is used increasingly for application of gene therapy. The mechanical particle delivery or gene gun methods to deliver DNA on microscopic particles into target tissue or cells. The process is increasingly used to introduce new genes into a range of bacterial, fungal plant, and mammalian species. It is the main method of choice for genetic engineering of many plant species including rice, corn, wheat, cotton, and soya bean. Presently, more than 50 types of genetically engineered plant species called the transgenic plants have been successfully developed. These plants were made to resist insect pests, viruses, or herbicides through the incorporation of foreign gene into DNA of host plant cells. Initially, transgenic plants were developed more in dicotyledons, but now extended to several monocotyledons like wheat, maize, rice, and oats. The transgenic plants have also been developed and are suitable for food industries, delaying the ripening in tomato. In gene farming, the use of transgenic plants as bioreactors or factories for production of specialty chemicals and pharmaceuticals is being pursued by a number of firms. Plants have been engineered to produce human proteins such as hormones in their seeds. A weed called mouse-eared cress has been engineered to produce a biodegradable plastic, that is, polyhydroxybutyrate or PHB in tissue granules. Mention some of the transgenic dicotyledonous plants. They are Nicotiana tabacum, Beta vulgaris, Glycine max, Helianthus annus, Solanum tuberosum, Gossypium hirsutum. What are the transgenic monocotyledonous plants? Asparagus species, Oryza sativa, Z. maize,